This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 841, recorded on December 10th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent and everybody else. I look out my window. I can see the George Washington Bridge from here. There are patches of blue sky with wisps of white clouds. It's a little coolish, obviously. It's almost winter. And indeed, it's, about 42 degrees or something like that?
I'll show you my view, Dixon. I have a camera on the windows.
Well, I can't actually see you now because I've got the screen filled with the show notes, unfortunately.
There's my view. The two windows are now full of...
you told me that you boarded them up.
Boarded up, waterproofing phone. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody.
Hey,
the weather here is nuts.
Really?
It's 84 degrees and sunny.
God. Oh, my God.
And tomorrow the high is going to be 59, and the overnight low tomorrow is forecast to be 33.
Exciting. That's exciting.
So something's going on.
Any snow on the way?
Oh, no. So, we had the snowpocalypse last year that nobody
It's true.
foretold. So who knows?
Right.
Also joining us today from New York, Amy Rosenfeld. Welcome.
Hello. Hello.
How does it look out your window there?
My window's full of viral snowflakes.
She can see my house from there.
I can, probably, yeah.
Oh, you put all the snowflakes in the window?
Well, that's where they're going. So
Nice.
They arrived this afternoon, and so in full glitter. So here's Heartland, and then...
Oh, it's got a heart in it. How cute.
Yeah, and then this, I think, is Lhasa. And then we have hepatitis C with the lipid droplets associated with it here. Oh, wait, now they're all tangled. And then we have the big final finale of the neutralized coronavirus. So, it's again all tangled. So here you see the neutralized coronavirus, and here's the antibody.
Nice.
And they were all sent to me and beautifully made by Jen. So we'd like to thank Jen, who's a listener and takes Vincent's online virology course and writes in during the live stream on Wednesday night. And she's a pediatric nurse in Rhode Island. And so
cool.
Now I'm all full of glitter, which is exactly what I like.
She sent you a list of what each one was, right?
Yeah, she sent the whole document. So here you can line them all up.
Oh my gosh. That's great.
And then she's an artist on her spare time. And so then she painted and made them all and painted them in glitter and sent me lights so that I could have them clipped through the windows. So the windows aren't washed here. So they're a little dreary. So it's good. It's excellent.
Very good. Excellent. All right. We have a lot to cover today. So let's get with it. We have two very brief reports about Omicron, which have just popped out in the last couple of days. There are actually more, but this is enough for today. First one is a medRxiv preprint. SARS-CoV-2 Omicron has extensive but incomplete escape of Pfizer BNT162b2 elicited neutralization and requires ACE2 for infection. The ACE2 part I'm not really so interested in, but the neutralization, let's take a look at that. This comes from laboratories mostly in South Africa, and Alex Siegel, Tulio de Oliveira, Penny Moore, first author, Sandil Saleh, and labs elsewhere, US, Germany. But what they've done here is to say, what about sera from people who received the Pfizer vaccine? How does it do in neutralizing Omicron? So they're using authentic SARS-CoV-2 virus and they have a neutralization assay. It's not a plaque assay, unfortunately. It's a focus assay because they say it's easier, to which I say easy should not be the reason you do something. But, it's not my experiment. So what you do here is you mix dilutions of antisera with virus, and then you infect cells and you ask what happens. And I'm gonna put this up because I think when I do the video, I will. This will be a cue for me to stick in this image. So this is really the main figure, figure 1, ACE2 partial neutralization of the variant by Pfizer. So each of these lines, well, they really shouldn't have lines, but they should just have dots, frankly, technically, because lines means there's a continuity between two results. And these are two viruses on the x-axis, the ancestral D614G and Omicron. And then on the y-axis, it's their 50% neutralization titer. So against D614G virus, so we have very good neutralization, either by sera from people who got 2 doses of Pfizer, and I guess this is the 3-4 week spacing here. Those are the red dots. So, 50% titer, about 1,000 or so at the maximum. It ranges from 100 to 1,000. And then if you do that with Omicron, then it takes a hit. Some of the people, each of these dots is a person. Some of them are below the limit of detection, and a couple are either at it or just above it. So not a lot of neutralization by those sera. Now, yes, go ahead, Amy.
So what does that mean in terms of protection? There's no correlation.
Well, we don't know. That's right. But,
right. So there's no correlation.
But the thing is, if you have 0 neutralization, can you even say anything about that? Because all you know is in your assay it's 0, but it might be in vivo. Okay, right?
Exactly. And it also depends, some people, it's like a plaque assay. Some people would never do anything beneath a certain level. Like Rich has talked about this previously, about how he doesn't plaque out undiluted.
Okay, so then we have green dots, which are people that have, let's see, they recovered from infection and have a dose of vaccine. Is that correct? Let me make sure I get that right.
Yes.
Okay. And
I don't know if it's, I'm not sure whether it's 1 or 2 doses, but infected and vaccinated, and we understand from other studies that infected in 1 dose, we did this paper last time, should be sufficient to give you superpowers.
So those sera do better against D614G. Now the 50% inhibition goes from about 10,000 to 100,000. And then now you have more neutralization of Omicron. You now go from about 1,000 to 10,000 or so. So that scenario, infection, vaccinated, you do better in terms of neutralization. Now, we don't know what any of this means, as Amy says, because we know that T cells are important for protecting against disease and hospitalization and death, and it's likely that they will do the same for this virus.
Well, the only thing you can extrapolate, I guess, to the real world is that in South Africa, the majority of people who have Omicron are recovered patients from Delta.
Is that right?
Mm-hmm. It's what Scott Gottlieb has said, and stuff.
So you and I were talking about this.
So that would suggest
tell us what it means, yeah.
That would suggest that prior infection leads to a vast breadth because it's a quasi-species, and in part there, within that antibody repertoire, there's an antibody that goes against Omicron, right? And that secondary infections are always going to be less severe. Although he then shot himself in the foot by saying that we should never make a Delta-specific vaccine because then you would only have immunity against Delta because that immunity is so very different from an immunity that is engineered against or elicited against any other variant. So it's very hard to know now.
Well, what I find funny is over on Med Twitter, people are taking the 50% neutralization titers and they have some secret sauce formula for calculating vaccine efficiency or efficacy. Can you believe that?
Med Twitter, isn't that a new journal?
I hear that there's a new editor-in-chief.
Yeah, I believe she's a friend of yours.
Really?
Yeah, Med Twitter is the latest journal. That's right.
Yeah, it's the latest journal. I wonder if you can cite it in your NIH biosketches. I have a publication there.
Don't do it.
I don't think so. Anyway, so that's the result. Other people are pumping out similar results.
So I want to point out that for the listeners that this particular article, I think, was the first of the things to hit the regular news, okay, where it said that the Omicron variant was over 40-fold less sensitive to the pre-existing immunity or immunity induced by ancestral viruses. And I just want to make the point that these are the data, okay? And when you hear reports like this, you can go look this up. I had to dig around. When the reports first came out, there were some news agencies that actually linked the studies. Others didn't. And I'm just want to communicate process here. You can, when you see a study or when you see a declaration in the news, you can dig around, okay? And I had to go to 2 or 3 news outlets until I found one that actually linked to this study, and I looked at the study because I wanted to see how many patients are we talking about, how were the tests done, and in particular, listeners to TWiV, I think, are probably clued in enough so that they can look this up and look at it and see what the data actually are.
Yep. I also want to point out that I don't know how far after immunization these data are, but at some point, these titers are going to contract, right? And you're not going to have as high a neutralization titer. So then the real question is, as Daniel said yesterday, what's the disease profile? That's what we need to know. In vaccinated people by whatever regimen, do we see 90% protection against severe disease or what? And we're not going to know that for a bit.
Yeah, that's the other thing is that when Omicron first showed up and there are news reports, I hear the newscasters and even public health officials saying, well, it's going to be at least a couple of weeks before we know. No, it's going to be months, okay? Because there has to be sufficient spread to understand something about the epidemiology and the dynamics, and there's got to be a lot of laboratory work to figure out whether this virus has any significant differences from pre-existing viruses. So no, not weeks, months.
Well, I mean, it was actually declared that it was more transmissible and less virulent.
Already.
Yeah. I mean, I don't know what, maybe I'm listening to the wrong news outlets or something rather. But as I said, it turns out that the majority of patients were previously Delta infected. So what that means is not really clear, especially when you shoot yourself in the foot by saying, well, the immunity that's generated or elicited against Delta is very different than the immune profile that's elicited against any other variant. And actually, if you look at some of the data, it looks like this variant is more like a beta, like the beta lineage.
Well, we were discussing this earlier, Amy, so yesterday Daniel reviewed a preprint saying that Omicron, what was the data now? I've forgotten. It's infecting people who have already been infected with Delta, but people who are not infected with Delta, it's less able to infect them. It's because, and you said this is because the immune response to Delta is so different from that of Omicron, right?
Well, it doesn't make any sense, right?
No, it doesn't make any sense.
So, I mean, I don't know how Gottlieb got to that conclusion, but it doesn't make. I mean, as I said, he shot himself in the foot when he said Delta was unable.
Like somebody mixed up the data.
Yeah, but it makes sense when you say that Omicron and people who got infected with Omicron had less severe disease, and then you find out, and they tell you the truth, that the majority of patients that they looked at were patients who were infected with Delta prior. The other thing is like they're only sequencing, at least in South Africa, they're only selectively sequencing people who had the spike dropout PCR positive test. And that's how they're determining whether or not you have Omicron or not. But they're, again, so it says, oh, we have 50 cases of Omicron. Yeah, well, if you sequenced only 50 people because they had the spike dropout, then you only had 50 cases of Omicron and you don't see anything else.
Wait a minute.
If you
Amy,
sequence...
there's a flaw in that, though, because in order to know whether the piece has been dropped out or not, you'd have to sequence everybody to find out who those other people were. So they have a lot more people sequenced than they're talking
No,
about.
actually, it's a PCR. It's not a sequence. They do a PCR. They look for the spike dropout of the PCR. And then they sequence those samples that had this dropout band of the PCR. I never like dropout bands of PCRs because you don't know if it dropped out or the reaction just didn't work. Like I forgot to add TAC, right?
Yes. Well, you have to have some controls, obviously, right?
Too many caveats.
Well, it's never good to have a dropout band, right?
I don't think so.
So,
No.
But you do have to agree that it's called the S something dropout, right?
Yeah, it's S dropout, yeah.
Right.
But eventually they remake the PCR primers and then it's no longer a dropout, right?
I don't think that they have if they're still calling it the S dropout.
Well, that's not good. It takes a day to make new primers, right?
I don't know.
All right,
I'm not
anyway,
good at the regulatory steps.
The other Omicron data is in the form of a press release from Pfizer. And one of our lit followers, Jeff, texted me this figure, which has the Pfizer BioNTech logo on it. 3 doses of BNT162b2 neutralize Omicron. So it's a neutralization assay. And we have sera from people who got 2 doses, and this is taken 21 days after the second dose. I presume these are stored sera from the clinical trials.
Yeah, I was thinking about this. These developers and all the labs that are doing this probably have lots of sera that they can pull out and do these sorts of things with immediately.
Yeah, but you don't want to keep freeze thawing it because that cracks the antibodies, and then you don't get the right titer.
Hopefully, they aliquoted it, right? Oh, that's not a verb. They made it into aliquots. Yeah, I'm sorry. They put the serum in aliquots. I would say maybe 50 microliter aliquots. Would that be good, Amy?
It could be. I mean, I don't know how much serum that they have.
Yeah,
I don't
if
know.
You had 5 mils, that would be a lot of aliquots.
It would be a lot of aliquots. I don't know. I don't know how they do. I don't know enough about what they do.
So let me put up this figure here. So this, on the left 3, we have, again, 2 doses of Pfizer. I think these are the compressed, the 3-week between doses. And then we have neutralization against the Wuhan ancestral virus, which, it's about 100 in this assay. And then Omicron goes below the level of detection. And then we have beta variant, which is between the two. It's better than Omicron, but not as good as the ancestral virus. But then on the right, we have 4 bars, and these are a 3rd dose a month laid after the 3rd dose. And I guess the 3rd dose was probably at least a few months after the 2nd. And now all the titers are quite similar. The neutralization titers, they're all slightly above 100. The highest are Wuhan and the Delta variant, and then Omicron and Beta are sort of similar in the titers. So the 3rd dose seems to have improved neutralization titers against Omicron, which is consistent with the paper we just discussed earlier this week, right, Rich?
Yeah, completely consistent. I see 2 phenomena going on here. One is that there is, with respect to neutralization titers, there is an effect of the boost. Okay, the increase in quantity, and secondly, that it. I don't know quite how to say this, but it sort of broadens the profile. You get a better, a more even distribution of activity against a variety of different variants. So I guess this is. Let me see. No, this is testing beta as well. So my sense is that if after your initial immunization, 2 doses, your immune system has a chance to rest and your memory B cells have a chance to go to school. Then when you then reimmunize, you boost and quantitatively those cells. And in the meantime, they have qualitatively increased. So they have a broader spectrum of activity. And that's completely consistent with what we saw before. And in fact, it's that consistency with the paper that we did last week that gives me some additional confidence that this is for real.
No, I think I would have liked to have seen serum from the first as a negative control, because if that serum is underneath 6, then you can make the argument that 6 is actually sufficient to supply some kind of protection.
Sorry, what do you mean, Amy? After the first dose only?
Yeah, after the first dose only, before the second dose. Because if it's under 6...
What's 6? Oh,
so
6,
if it's under 6,
okay,
then you could conclude that you had little. Then you could conclude you have a little protection or some protection, versus here where they're trying to say that you have like no protection. So it would have been better if they had, after the first dose, because you claim that the first dose didn't give you a high enough efficacy against protection of disease, right?
Right, that's correct. So we have a mix now of people who have 2 doses and 3, so it'll be interesting to see going forward what Omicron does in them in terms of disease, right? We can compare those 2 groups.
Actually, I can, this is somewhat peripheral, but as I was saying last week, I had my second blood draw for an antibody test for me after having a boost. Now, once again, in this particular case, the quantification is even less accurate, I guess you would say. But between my first blood draw and my second blood draw, it was about 3 months. The quantitative result has increased with a boost in between. So that all.
Yeah, but it would have been actually nice to have had a blood draw right before you got boosted.
Yes, but I don't have any control over that, and we'll see what happens. Yes, I would have liked that too.
And it would, because you want to know if the difference is the same, and you're just moving the starting point of the graph to if the slope of the line is the same, or you're just moving the starting point up the y-axis, so you're kind of like not really changing anything.
My wife may actually do that control. She hasn't been boosted yet, and we're anticipating she's going to get her second draw.
Good.
I will report back later. I also want to, I got a couple of things. First of all, quoted directly from this Pfizer press release, quote, "It's clear from these preliminary data," referring to the data we just discussed, "that protection is improved with a 3rd dose of our vaccine." No. Okay. So I just want to make that point, because everybody, I think this is a casual error in a way. I think that but this is a problem, because these casual errors become fixed in the public mentality, and that's what leads to the misunderstanding of what's going on. I have to believe that if you pinned down Pfizer on this, they would say, oh, yeah, sorry, we didn't really mean protection. But you got to be careful.
Seriously?
I don't know.
You really think that they're going to not say that they meant protection?
I don't know. I don't know.
Yeah, you can't say that, but yeah, it may be casual, but it's just neutralization titer. We don't have a correlation, as you will see. We're going to do a paper in a bit.
So then I want to ask. So we keep hammering on this, that these sorts of data don't equate to protection. But so why do we even do the experiments? What does this mean? Okay, if it doesn't, what value do these experiments have? Can we? I mean, do you? What does it mean to you that after the third dose of the Pfizer vaccine in this assay, the titers against Omicron go up?
Mm-hmm.
Does that have any?
I do have a thought about that. If you
go for it.
So, if you are, and Amy, just wait till I'm done before you say.
Why are you yelling at me already? I haven't done anything.
Because I talk to you a lot. Just wait. So, when you get the third dose in the next 6 months, you're going to have relatively high serum antibody titers against spike, and if you were infected, you're going to neutralize the virus as it enters your nasopharyngeal cavity, and it's not going to get very far. So you probably won't have even mild disease. But as these antibody levels contract, then if you're exposed to virus, you will have a couple of days of it reproducing before the memory response kicks in. And then you may have less than moderate symptoms or more than mild symptoms, say. But if those antibodies better neutralize Omicron, then that memory response will be able to control it better before it gets to the point where you really depend on the T cells to keep you out of the hospital. That's how I'm looking at it. I know it's a hypothesis, but that's why I think these are valuable, because if you make antibodies and the memory and they barely neutralize Omicron, then that could be a problem. You might get more severe disease.
Really? You want to? I was going to say you want to negate FC receptor mediated.
Well, no. I don't want to negate it. No.
Of mediated by the antibodies and stuff.
I don't want to negate it. Well, we don't have evidence for it for SARS-CoV-2 yet, right?
No, there is a paper that talks about FC receptor mediated.
I know it can happen. But in these studies, they never measure that in any of these functional studies.
Well, these studies also only measure IgG, and you and Fauci and various other people have come out and said that there's no IgG in the upper respiratory tract and in the nasal pharynx. So I don't care about IgG in the lungs if you're arguing now it's going to protect me against mild infection and early infection, because those events occur in the nasopharyngeal cavity.
It's all about the nasopharynx.
Right. So this is measuring IgG, and that cavity, that memory, as you will read, is dependent upon IgA.
Agreed. I agree.
So
hypothesis. It's just a hypothesis.
Right. But even still, you're still only measuring IgG.
In the serum, yeah. Right.
So if you actually, and Theodora and Paul have a paper that was back-to-back with another group about the importance of dimeric IgA. Mm-hmm. And if you talk to the person across the street, he will tell you we need to make an IgA vaccine.
So basically you think these data are useless?
No, I didn't say that they aren't useless. I'm just saying I'm not basing any hypothesis on them.
Okay.
Because the infection doesn't occur in my lungs.
I
it
agree.
Occurs in my nasopharyngeal cavity.
Well, it starts in the nasopharynx, and if you can clear it there, it won't get to the lung, right? That's the idea.
And so I think that maybe if you put a gun to my head, maybe I would stretch a hypothesis that this is protecting from only severe disease because that's in the lung, and this is where that antibody is present.
So,
but
Amy, what does the Regeneron monoclonal antibody do?
It's got to protect against severe disease because it's IgG.
Yeah, but that's not in the nasopharynx that they're giving it.
They're giving it systemically, and it clears it. And you have to have some type of severe symptom. You can't just go and say, I've been exposed.
But it's not in the nasopharynx. It's in the main systemic.
Prevent severe disease, yeah.
Yeah.
Right. So what kind of vaccine do you going to, what kind of a vaccine can you think of that would only elicit IgA?
There isn't any.
I know.
Or actually, there is. There is. Actually, the intranasal flu vaccine would elicit IgA.
Anyway, Rich, that's what I think.
Yeah. So I guess the thrust of my question here is that in hammering always on saying that you can't draw conclusions about protection from these sorts of experiments, well, I'm left with a question: what can you conclude what utility do these experiments have. And what I'm hearing you say is that despite the fact that you can't necessarily make clear statements about protection from this, in your mind, these do have some sort of physiological relevance.
I think so, but Amy's points were absolutely valid that it's much more than IgG neutralization and other things going on.
Yeah, but
I think at the end of the day,
if you want to prevent severe disease, which is what their vaccine claims to primarily focus on, then IgG is a perfectly logical antibody to look at because you don't want it to become systemic. You can become infected and have a mild infection, but once it becomes systemic, it becomes a big mess of cytokine storms and all those other things.
It doesn't really become systemic. It just goes down into the lung, right?
Well, yes, but IgG plays a role.
Sure.
That's what happens.
Yeah, but IgG can cross the epithelial barrier. Yeah.
Yeah, but I think probably the safest thing to say is that you have some kind of immunogenic response.
Yeah, that's
which is you can't say anything about protection, disease, blah blah. What you can say is you can elicit you have in it a potent immunogen, and what that means. Take your pick of anything that you want it to mean, but that's basically all you can say. Whereas if you took inactivated virus and you got nothing, you would say you killed the immunogen, basically. How's that for you, Richard?
Yeah, that's kind of where I was going. That's it's minimally these sorts of data are a good measure of that your immune system's doing something. Okay.
Yeah, if the
titers go up, that probably means you've gotten an immune response. If the titers give you a different profile of neutralization with different variants, that's some indication of the maturation of the immune response. But you don't necessarily know how that translates to protection until, how that translates to protection until we see the epidemiology.
Well, I'm not even sure that I would say that it says anything about variants because you can, I know that the paper that you did on Tuesday, because I picked it, had Biomarkers measurements and everything, but this doesn't have any Biomarkers measurements. So I could make the argument that you're making a lot of weakly binding stuff, but because you've made so much of it, it now has high avidity.
Right. Yeah, it could be. Sure.
Right?
Yeah.
So you really you really can't say you have gone through, matured the immune response and done hypersomatic mutation and all this other stuff that I don't really know anything about.
No.
Yeah, that I don't really know anything about. But
Dixon, what were you going to say?
I was just going to say, even if the titers were low, it didn't mean that those people weren't protected because we fall back on the T-cell interaction system. So
absolutely,
it's not depressing to see that they're not responding as well as they should, given the fact that there's another branch to the immune system that's going to be active as well.
But there's also other ways that antibodies function.
We're going to learn about that today, right?
Yeah, this is
hopefully because I picked this paper too.
You did. All of them.
Are you being led around by the nose?
I read a lot.
No pun intended. No pun intended on that one, by the way.
No, she doesn't say you have to do these. She says this is interesting, and I say yeah.
Absolutely.
But let's move on to our snippet, which is a science paper. Impact of community masking on COVID-19, a cluster randomized trial in Bangladesh. Very interesting. So we have Jason Abaluk, we have Laura Kwong and Ashley Staszynski as the first three co-authors, and then the corresponding author is Ahmed Mushfiq Mubarak. And these are from all over, Yale University, Stanford, Bangladesh, of course, several places, Australia. And this is a very interesting study that we've been wanting. Face masks have been contentious, and they point out here, which is interesting, that WHO did not recommend using face masks until June 2020, and they said there's no evidence from trials that they work, and they worried that when people wore masks, they would abandon other safe behavior. So those two issues they addressed in this trial. Very interesting. It's not easy to do a randomized controlled trials. Think about it. You tell people to wear a face mask. How do you know they're going to wear it all the time? I mean, they did
observe.
They did do observations when people go in their homes, when they go. I mean, I just have not a lot of faith that people are going to do what you ask of them all the time. Anyway, this is in Bangladesh. It's just, they had at their peak 15,000 cases a day, 165 million people in Bangladesh. And they say the number of cases and deaths are probably underreported by 1 to 2 orders of magnitude, 10 to 100 times more than they're actually reporting. The government formally mandated mask use in May of 2020, but they say enforcement was weak to non-existent. So in this trial, let's see, they had 600 villages throughout Bangladesh. There's a map, and you can see they're all over the place. 125,000 households and 342,000 individuals. And some of them got to wear masks and the others did not. They were the control group. And what is cool about this study is that they tested people by blood, taking blood and looking for antibodies to the virus to confirm infection. So symptomatic seroprevalence, that's the primary outcome. So people have symptoms and then they go and they get their blood tested, which is nice. It's very nice to do that. So the mask wearing was 13% in the control villages and 42% in the treatment villages.
My goodness.
Yeah, that's not bad, right? These are the people that they enrolled in the trial.
And they in the test villages that were masked, the way they accomplished that was with a bunch of publicity and also distributing masks. Okay, so you could get masks, and I got the impression from the way it was written that you get a lot of people who respond positively to being given a mask. Oh yeah, I want a mask.
Sure.
But still, it's only 42%. Oh, and then as has already been alluded to, how do you know what the compliance was? Well, they sent out observers, okay, who in some fashion or another tried to make an assessment of what the compliance was. I presume they also surveyed participants, said, did you wear your mask? So a little of both.
The data,
but these are hard data to get.
But look at the result. It meant that a lot of them were compliant.
Oh, yeah, it made a difference.
Huge.
All right, so...
that's the proof is in the pudding.
Where the heck is the numbers here? I've lost the symptomatic results.
If you go to your blog, you summarized it.
Yeah, maybe I should just look at my blog post. That's a good idea.
I'm helpful.
All right, wearing masks reduced symptomatic seroprevalence in treatment villages 0.68% compared to control villages 0.76%. So that's the fraction of the study population that got reported symptoms and were tested to be positive by serology. So the reduction is 11% by mask wearing. But what's even perhaps more interesting, 23% between the ages of 50 and 60 and 35% in people over 60 years old. The reduction in seroprevalence. So that means 1,514 fewer people reporting symptoms in the mask groups and 105 fewer seropositive. And so, why do older people have a greater reduction? They don't know. They didn't figure that out.
I wonder if the older people weren't more really not only vigilant about their mask use, diligent about their mask use, but more honest about whether or not they used the mask.
They didn't get that much compared to the younger.
Oh, that's a good point too. Yes.
Well, they adjust them all as potential.
Yeah, they said they
reasons.
might not have gone out much, and the other thing is maybe it's easier to remove droplets for older people in a mask. So in other words, perhaps you need, what is it that they say? They say the mask has a better chance of removing an infectious droplet in an older people than a young person because the dose is different, or I'm not sure what they meant by that. Anyway, so those numbers don't sound like a lot, right? But they point out that this does not mean that masking prevents only 10% of cases or deaths, because they didn't mask everyone. And they said it's likely that near universal masking would have an even greater impact.
Right, much greater impact.
Right, but it also showed, which is one of the more important parts, that actually if you wore a mask, you are more likely to physically distance.
That's
I was
right.
going to just point that out also. You're taking it serious when you put on a mask. You think you're doing something to prevent the infection, so you might as well follow the rest of the rules also.
Yeah, and like they saw differences in the market versus the mosque and everything and stuff.
Sure.
And I think they come out with some economic calculation about how much at the end of this saved the economy, which is the opposite of the argument here.
It said that we estimate that a scaled version of our intervention being implemented in Bangladesh will cost about $1.50 a person, and between 10,000 and 52,000 per life saved.
Right, which is as I said, it's the opposite philosophy of the nonconformers here, right?
In the U.S.?
Yeah. Don't wear a mask because, it's going to cost the economy more because you're not going to do X, Y, and Z. And we should open the economy and not have people wear masks because it's not so aesthetically pleasing, or it mongers fear, or whatever it was, right?
The other little tidbit in this is that I don't know how significant, I didn't look into the statistical significance, but reproducibly, surgical masks seemed at least a little bit better than cloth masks, which I thought was interesting. I've sort of, my own personal experience is that I've come to kind of like the surgical masks better than all the cloth masks that I've tried anyway.
I agree with you.
Why do you like them better? What about it?
Among other things, they clip around my nose. Okay, which I like, and they're frankly easier to breathe through.
They are
than some of the cloth masks. It all makes sense. I mean, they were designed for this, right?
Exactly.
Yeah, of course. Do you have less fogging of your glasses, Rich?
Yeah, because I can pinch them over my nose.
Yeah, because I wear a cloth, and the fogging, especially now in winter, is really bad.
Yeah, that's right.
Amy, you wear a surgical mask, right?
Yes.
So
but I don't wear glasses.
But do you guys use a new one every day, or
no?
No,
I recycle my surgical masks. I'm the only one who uses it. Reminds me of that cartoon early on in the pandemic where the mother has her kid coming home from school, and she notices he's wearing a different mask than he went to school with, and he says, "Oh well, Johnny and I traded."
That's good
because he liked mine.
Yeah. So the other last point from this, so we have a controlled trial that shows they work. We have information that people don't decrease their physical distancing, right? Which are the 2 points of the WHO. And the final thing, which I think is cool, is they say, so the study is over and we still observed for 10 weeks and mask wearing didn't drop, even though we said the study's over. People kept wearing masks.
That's right.
And then after that, it dropped, but they were still 10 percentage points higher in the treatment areas in terms of wearing masks. So I guess you get people used to wearing masks and they continue, right?
I was going to say, that's my experience. I get used to wearing a mask and I continue. As a matter of fact, it feels a little funny to go into a store without a mask.
Oh, I agree with you. Totally.
You know what I was thinking the other day? Masks work. And so when you don't wear them, you're breathing in other people's breath.
Really? You just...
Yeah, it just hit me,
Revelation.
To the extent of which you breathe in other people's breath is huge, and that's why this is transmitting, right?
Yep.
But I never thought of it.
Well, I don't know. How did you think flu got transmitted or rhino.
Obviously, I knew this, but for me personally, sitting in a train, right? Now I'm protected against other people's breath. And maybe I'm going to wear this for a long time in crowded places. I don't know.
I know exactly what you mean. This is sort of coming to a new sort of insight or realization of something intellectually you have known all along. Yeah. But all of a sudden it strikes you, holy cow, right? In a new way.
Yeah, but he also has been known to follow up this with, when can we stop wearing masks because they're fogging up my glasses, or I don't like how they feel, or I'm just tired.
Yeah, I've been complaining, but I might, in certain situations, continue. And I might also wear them when I'm in a bathroom. In a public bathroom, because, the toilets and urinals make aerosols. And so a mask would be great to prevent that. People have, polyomaviruses in their urine. I don't want to breathe that in.
I think at this point we've all been exposed. I think,
I understand.
I think you probably have a robust immune response at this point. But I think it's actually an interesting concept in order to counter, severe flu or in RSV seasons, right?
As Rich said, it was kind of just an awakening for me, because I didn't think about it much. And now you wear a mask every day, you think about it. Man, I've been breathing a lot of people's breath for 69 years, And,
yeah, I'm just teasing you. It's okay.
And actually, most of us are okay. That's the amazing part, right?
That's herd immunity for you.
Yeah, right. All right, now we have a paper today published in Cell Host and Microbe, which is about respiratory syncytial virus. And it's another eye-opener. Okay, the masks is one kind of eye-opener. And this, for me, now I realize when you say correlative protection, man, you're not just talking about antibody. Holy cow, or T cells. It's a lot of other stuff. And this really helped me appreciate that. So the paper is upper and lower respiratory tract correlates of protection against respiratory syncytial virus following vaccination of non-human primates. And already you can see we should be talking about upper and lower tract, not just correlates. This comes from the Reagan Institute of Mass General, MIT, and Harvard, and Merck in Kenilworth, New Jersey. Wow, Merck in Kenilworth, New Jersey. And the lead, the corresponding author is Andrew Bett and Galit Alter, the last two authors. And then we have the first author is Tomer Zohar. So this has a great summary of respiratory syncytial virus. And again, Amy, I have to apologize for saying it doesn't do much in older people. Apparently, older adults represent another vulnerable population, accounting for nearly 200,000 infections and 10,000 deaths a year in the U.S. alone. And she told me this the other day. I said, no, you're wrong. You're wrong. And she's right. God damn it, she's right.
Basically, I'm not clear why we don't have a podcast or you don't have a book that just says the Amy Wright pandemic. But it's
a big pathogen of newborns, right? 33.
I wasn't negating the newborn thing.
New infections annually, highest mortalities in developing countries. Most children become infected their first few years of life. Most cases are mild, but a certain fraction go on to develop bronchiolitis, pneumonia, and broad inflammation of the respiratory tract. The most common cause of childhood lower respiratory tract infections accounts for a third of the deaths associated with those in the first year of life. And what do we do about it? Well, we have monoclonals, but they're mostly used for premature infants. They're incredibly expensive.
Premature infants at risk. Yeah.
There was an experimental vaccine produced in the 60s, which led to even more severe disease. So that was abandoned. And as Barney Graham once told me at a meeting in January 2020, we spent 50 years afraid to make new vaccines, trying to figure out what we did wrong. But they say, vaccine development has failed in part because we have a limited understanding of the correlates of immunity required. And this is a perfect way to put it. The correlates of immunity needed to prevent infection and disease. We always say correlates of protection, but I guess that's not right. We should specify. correlates of immunity for infection and disease. So what do we know about RSV? So antibodies may be enough. Monoclonal antibodies are if you treat people, you can reduce hospitalizations. Neutralizing antibodies appear to be a predictor of protection against disease. And they say, though, prediction does not imply mechanistic causality. So these are some of the issues we're going to get into here. So there's no licensed vaccine. And they say this is probably in part because we have a strict focus on antibody titers and neutralizing antibodies. And that's probably not right.
Barking up the wrong tree.
It's not just about neutralizing antibodies, but maybe non-neutralizing antibody functions in protection against not just RSV, but other diseases as well. But I guess we do have a real focus on neutralizing antibodies, as shown by the previous 45 minutes discussion, right, of the 2 Omicron reports. And then they go on to talk about FC-mediated activity of antibodies. So the FC portion of the antibody, the part away from the antigen binding site, they can bind to receptors on various cells like NK cells, which can then mediate protection. So an NK cell will recognize an antibody bound to an infected cell and kill it. And there's been some evidence that these are important for protection against respiratory syncytial virus. They can reduce viral loads. So this is what they want to address in this paper, where they have an amazing set of viruses to use. They use non-human primates, which I didn't realize are only a semi-permissive host. So you're just looking at infection in these animals. They don't develop much of disease. But they have 6 different vaccine strategies. And each one, there's some different vaccines and some different ways of administering them, as you'll see. Each one gives you a different level of protection against infection, a different antibody profile, and a different profile of these other functions, these FC-mediated functions. And so they do these experiments, and then they use algorithms to say what's the best, which ones do the best. And I'm thinking, I don't know about you, Rich, but I'm thinking, in the old days, I would just look at the data and say, yeah, that's the best one.
Yeah.
But I guess it's too complicated now. You need to use software.
It's exquisitely complicated.
And you too, Dixon, right? You'd look at the data and say, yeah, I can see the pattern there, but you can't do that anymore. All right. So what are the vaccines? These are very interesting. So African green monkeys are the vervet monkeys, I think is the right word, AGMs. They got one of 6 different vaccines, and each of them give you the F protein of RSV, the fusion protein. So it's not the spike equivalent, the receptor binding, it's the fusion protein equivalent. And I think that's part of the problem with that vaccine that failed, is that the F protein was destroyed by formalin treatment.
We probably ought to just briefly say that this is a negative strand RNA virus that has 2 surface proteins, F and G, right? G is the binding protein, F is the fusion protein. I don't myself know why the focus is so heavy on F. Maybe that's the immunodominant epitope in an immune response, but I don't know.
Well, I think part of the reason is if you just make antibodies to the G, the glycoprotein G, right?
Right.
Apparently, the viruses can still travel cell to cell by fusing.
Oh, okay.
And they evade, so there's anti-G doesn't prevent that. And that's why, in part, I think those kids had serious disease.
All right.
So let me tell you what these different vaccine modalities did. So they're all, as I said, based on F protein. 3 of the groups were given different formulations of a pre-fusion stabilized F protein base, so purified protein, but the protein is pre-fusion stabilized, as we see what was done for the spike of SARS-CoV-2. And these are called DSCAV1. 2 of these 3 were given intranasally in what they call a 20% nano emulsion. That's an adjuvant that's good at inducing mucosal immunity. And that's good to know. I didn't realize that you could have an adjuvant that did that. And they do 2 different doses, 0.1 ml and 0.25 ml, the same amount of protein. And they say the higher dose delivers the vaccine to the lower tract in comparison with the low dose, which it retains in the nasal cavity. So I guess it drains down, right? When you spray so much volume in the
I guess
nose.
so.
The third one is given intramuscularly with an aluminum phosphate adjuvant, again, in 1 volume, 1 dose. 2 doses were given full-length post-fusion F protein nanoparticle vaccines intramuscularly. And then the final group was given an mRNA lipid nanoparticle vaccine, which encodes the F protein.
Intramuscularly.
Yeah.
And the other 2, the post-fusion guys, they had, as you said, 2 different ones. And the difference there is one is adjuvanted and the other is not.
That's right. So they have a control. So there are 2 controls. One is an empty lipid nanoparticle vaccine as a negative control, IM. And the second group was given a infectious attenuated strain of RSV intranasally as a positive control, showing that the animals got infected and so forth. So they give vaccine at 0, 29, 56 days. Then they challenge them with this attenuated vaccine. So that's the challenge. These are actually challenge studies. And they're challenged intranasally and intratracheally on day 70. So immunize, 3 doses, then challenge with this vaccine strain. So they're not using a wild type strain. They're using the vaccine strain.
I spent a lot of time on this virus, RSV-A2, because I'm looking at this and I'm thinking, why are we even messing with all this other stuff if there's this attenuated strain around?
Right. What's the story with this?
It is basically a laboratory strain. The best I could do in terms of understanding what it is a paper from Antiviral Research in 2004 that describes, now I don't know that all these things that are called A2 are the same, and I'm assuming for the time being that they are. They say that this was the inoculum that they're using in this paper was prepared from a virus stock initially isolated from a child in Melbourne, Australia in 1961, passes 6 times in human embryonic kidney cells, 9 times in primary calf kidney cells, 2 different temperatures, then cloned, several serial clonings in calf kidney cells, then passed 4 times in primary bovine kidney cells at 35 and 5 times in MRC cells at 32. So this is a pretty classic. I don't know that this was intentional to make an attenuated strain or simply that this was sort of the sloppy lab history of this particular virus. But that the idea is that it has a passage history in culture that has the effect of attenuating it relative to the original virus. And they know that from animal studies. And in fact, this virus has been used, since it is attenuated, in clinical trials as a challenge virus in humans. Okay? But I gather that it still causes some symptomatology. Okay? So you can get a mild upper respiratory infection from it, though its tendency to cause severe disease is less. And I presume, and I'm just presuming here, that that's one of probably many reasons why this is not actually being touted as a real vaccine. In this in this monkey model, you can, if you give the virus, and you can see this in the controls where they've not been immunized, if you give the virus, you can measure virus in the upper respiratory tract and in the lungs. So that way you can track an infection. But you can also it doesn't kill any of the animals. So you can give them this virus and let them rest for a while, and now they've been immunized. So that's the positive control for the immunization.
Got it. So what they do essentially is after the challenge, at different times they collect nasopharyngeal wash. They do a bronchoalveolar lavage. They put some saline down into the lung in a tube and they pull it out so you get a sampling of what's down there. And then they measure a variety of parameters. First, they measure infectious virus. They do plaque assays and measure PFU per mil. And then they will do immunological measurements as well. So the outcomes vary. There's one figure with all of these data in the one figure. And so that this attenuated virus, immunization with that completely suppressed reproduction of virus in the lung.
Right.
And the challenge is itself. So that does a pretty good job at blocking infection. Also, this one of these CAV1AD, I don't remember which one that is also very good at protecting the lung.
That's the to me this is relevant because so not only are we talking about all these different vaccines, but a critical part of this whole thing, maybe you've already said this, is that we're looking at two distinctly different compartments.
Yeah.
That I think there's a great tendency to not to think about the different compartments and conflate things. But the important thing is that we're looking at upper respiratory tract, that is sort of above your larynx, and lower respiratory tract, your lungs, and RSV and SARS-CoV-2, the serious disease results from what's going on in your lungs. So if an infection is restricted primarily to the upper respiratory tract, you're much better off. If it gets down into your lungs and is causing problems, you get more serious disease. And the point here is that, as we'll see, the immune response in those two compartments is different. And this is all relevant to SARS-CoV-2 as well as this. This is terrific. The dsCAV1AD, this is how I got off on this, because I think it makes a difference. That's the one that's the same immunogen, the dsCAV1, but it's injected, as opposed to the other two guys that are intranasal. So the route of immunization is going to make a difference.
Yeah.
Yeah.
So you can see. Go ahead. Sorry.
Well, we've known that. You and I have talked about this on TWiV with an Ebola paper and a SARS-CoV-2 paper prior to this, like in the summer.
And here they're comparing them directly, so you can see. And these different modalities had different effects on virus in the lung, and they're different from virus in the nasopharyngeal wash as well. The F protein, RSV-F, didn't work.
Right.
The only one that and in fact, that's the lung. In the nose, the only one that gave a complete control of viral loads was the attenuated virus. So the nose is very hard to prevent virus reproduction in.
Yeah, I think they'd say that almost everything did a little something, but not much. The thing that did the most in the nose was the mRNA vaccine. Likewise, in the lung, you already said that the injected CAV1 vaccine, protein vaccine, did a pretty good job. The only one, aside from the live attenuated control, that really wiped out infection of the lung, again, was the messenger RNA vaccine. So I'm biased here, but the messenger RNA vaccine is winning this race.
So they said RSV-A2, the attenuated, exhibited the best overall control, followed by MDS-CAV1, which is the
that's the messenger RNA guy.
It's the mRNA one, MDS-CAV1. Hold on. MDS-CAV1, yeah, the mRNA vaccine, right. And then dsCAV1AD, which is the intramuscular protein inoculation. highlighting a clear hierarchy in vaccine-induced protection and pointing to the successful induction of differential viral control across upper and lower tracts. So then they said, let's look at various immune parameters and see if we can make any correlates in the upper and lower tract. And they looked, of course, at neutralization in serum, but they looked at IgM, they looked at IgA, they looked at IgG, they looked at FC-mediated effects, like antibody-dependent phagocytosis, antibody-dependent complement deposition, NK cell degranulation, a whole lot of them, and they're all different. They're different depending on which of these vaccines that have been used, and so then you have to convolute all these data, right? You have to somehow say, okay, these are the protection patterns. What immunological parameters correlate with this in both the upper and the lower tract, right? So we got antibodies, antibody titers, FC receptor function, FC portion activities. We have IgA and IgG and IgM titers, and we have viral loads. So they have software that integrates all of this and says, gives you an answer, right? So the first conclusion is the antibody functions are more strongly linked to protection than neutralization. Functions, that means not neutralizing antibodies, but the functional activity. For example, in the nasal profiles, although few significant correlations were noted, although IgG titers, FC binding levels, and pre-fusion antibody titers were correlated protection at 1 day, day 3. So in the upper tract, protection of the nasal cavity, it's hard to pinpoint any one thing that really predominates. But they say in contrast, many features of vaccine-induced immunity were linked to enhanced control in the lung. You know, these antibody-dependent cellular functions and so forth. NK cells, FC binding, and so forth. So then they tried to determine the minimal humoral biomarkers of immunity against RSV, and this is where they use different software approaches to do this. Let's see if I can find a summary to this. Okay. I did too much highlighting here. Sorry, folks. Hold on. As few as 3 antibody features of the total 13 captured, so they had 3 different measurements of antibodies, were able to accurately separate animals with viral loads past the lower limit in the nasal cavity. The top features were all enriched to include complement deposition, pre-fusion IgA responses, and neutralizing antibody titers. So 3 different things are correlating with protection of the upper tract. Although it wasn't great, that's what came out of it. And at day 5, interestingly, IgM is very predictive of protection. And they say this is IgM. What does that do? It drives complement activation, right? So that can lyse virus-infected cells or even membrane-containing virus particles. So they say in the upper tract, strict neutralization with complementing activating activity may be enough to delay infection in the upper tract. So I think that's important. We're just thinking about neutralizing antibodies, but it's more, it's complement activation. In the lungs, at day 3, 3 features, antibody-dependent NK cell chemokine secretion, a very specific chemokine, complement deposition, and IgA, all correlated at day 3 with protection. But at day 5, there were 5 different things that correlated. I don't need to tell you what they all are. And the same thing at 7 days. Different immunological measurements correlated with protection. And so their summary is, they call this a holistic agnostic analysis. NK cells complement IgA and neutrophil phagocytosis may play a dominant role in containing RSV in the lower tract. The only thing there, and I guess the IgA is the only neutralizing activity. Everything else is non-neutralizing.
And yet,
and these,
most of our assays only look at IgG.
that's
Like
right.
if you look at,
And that's in the lower tract, and the
That's
upper
in the
tract
lower
is
tract.
different.
yeah.
Okay.
Upper tract is different. Yeah.
And they had before the last summary that you read, because we've been talking about neutralization tests. They say, although neutralization was not selected as a correlate of immunity by the models, that is, neutralization is one of the things they tested, and that didn't pop out as being one of the important things in their mathematical models. They say neutralization was, however, clearly associated with reduced viral loads by univariate analysis on day 3 in the bronchoalveolar lavage, suggesting that neutralization may play a supportive mechanistic role in the lung, particularly early on during infection. So that's a long-winded way of saying that neutralization may mean something, but it certainly doesn't pop out as the major factor.
The driver. That's
And
right.
yet,
So that's what we have to have in mind as we're looking at all of these other, of course, it's different. SARS-CoV-2 is a different virus, but this is the kind of thing we need to keep in mind as we're looking at all of these antibody tests, serum tests that come out when we're looking at Omicron variant and stuff.
well, any variant, but
Any variant, yes.
but yet we've based our entire public health policy on neutralization assays.
Yes, that's why this paper is eye-opening, I think.
Yeah.
Yeah. Of course, we didn't have time to do all of this, right?
Right.
Really? Because I'm still waiting for polio, and we've had a vaccine against polio for 50 years, and we have 2 vaccines, and one is oral and one is intramuscular, blah, blah, blah. So when do you think we'll get to that study?
Well, that's the other, one of the reasons that we do the measurements we do is because they're easy.
Yeah, that's right.
That's not to fault anybody who's doing them, because the hard tests are really hard. Okay, the antibody tests it's easy to get the material to do the test with. It's easy to devise tests that can even be mechanized. The other tests are much more difficult.
So let me summarize 2 more things here. First, okay, IgA titer is important in both compartments on all days. In both compartments, antibody-dependent complement deposition was day 3. IgM was day 5, and they say this drives complement deposition and also phagocytic clearance of particles in the lungs. Neutralizing antibodies were a marker of clearance early in the nasal cavity, but less apparent over time and not in the lower respiratory tract. Isn't that interesting? Just in the upper tract. So they say strict neutralization may only be key to protection early in the upper tract, and complement-mediated activity may be the key on day 3 across compartments as well as NK cells and neutrophils. So which vaccines are the good ones? Here's their conclusion. All biomarkers were robustly induced by the mRNA vaccine.
Let's hear it for messenger RNA. I love it.
And kind of, Amy says they did do the right thing in the end because the mRNA SARS-CoV-2 vaccines worked well. Maybe for reasons we don't even know. Maybe for reasons beyond neutralizing titers, right?
Yeah, I mean, I think that some of our conclusions, again, about whether or not the vaccine's efficacy is waning or effectiveness, whatever it is now, are a bit hasty.
Yeah.
And I think that prudence is always better, because when you put out a hasty statement and you have to retract it, it's not that easy. And sometimes I think, so sure, I'm all for transparency, but you have to know when to release the data. You have to know how to do it, and it, stuff. And
So,
I just don't think that it's. I just think that there's been some mistakes.
I think this, for me, it tells me just test an mRNA vaccine against respiratory syncytial virus, because if it's good at inducing all the right biomarkers, why not, right? Intramuscular mRNA. So a couple of quick comments from the discussion. They write, while antibodies represent a correlate of immunity measured in most licensed vaccine, that's true, antibody titers alone have been insufficient for providing insight into what we need to protect against many pathogens, including RSV. And now we know why, because other functions besides neutralization appear to be important. Maybe that's part of the HIV issue, right? I don't know. Let's see, what did they say at the very end? Yeah, they have some nice discussion about the upper and lower tract, that if you don't get protected in the upper tract, then neutralization doesn't matter as much in the lower tract. Their cellular functions are more important. I think this is RSV, but I wonder if it's a similar situation for COVID, right?
For any respiratory disease.
Any respiratory disease, yeah.
It's not,
Then they also say whether mRNA vaccination could lead to complete protection in the nasal cavity if given intranasally remains unclear. That's an interesting idea. Put the mRNA IN instead of IM. That should be tested. That's a cool idea. Also, a lot of good discussion here. They say we have to do more optimization here. But they say the mRNA vaccination may be the key.
Well, Vincent, it also raises a problem here, though, because they're using non-human primates, and the correlation between vervet monkeys and humans
good point.
may show some interesting differences in terms of all those biomarkers as to which vaccines are even more efficacious in humans than in monkeys.
Not only that, but their model virus is an attenuated virus.
Exactly.
That's right. I mean, all these
things. It's a great study to just shows, given lots of different choices as to how you configure your vaccination and the scheduling and the spacing and all those other things that come in, adjuvants and no adjuvants. I've dealt with those problems too in the past in my own research. And what does the FDA look for? FDA to approve a vaccine, they take whatever the company says are the good indicators that their vaccine is working, and they judge whether or not it induces those things. And they're saying neutralizing antibodies are what we can measure, and that's what we're going to show you.
Yeah, but it's just a correlate. It's not a mechanistic correlate.
No, I understand that, but how does the FDA approve of the vaccine other than going by the data that the company gives them?
That's all they can do, and they do safety and efficacy, right?
Right.
Against disease.
And efficacy is trial and error.
But you're right, Dixon. These may have no relationships to what we see in people.
But it's a great set of experiments, though. It's really true.
But the thing is, you can't do most of this in people, right? You can't get a BAL. You can't get 5 time points and measure virus.
No.
So what you do is you take this and maybe you tweak it a little more in non-human primates and find something that's better than mRNA, and then you have to try it in people, right?
Well, maybe what you do is you say these were the things that correlated with protection, and you make a very specific, very sensitive assay that somebody can do off of human samples easily. You don't have to look at all of them. You just have to say these were the top 3. So we can look at IgA, right?
We can.
So you look for, you take some scraping or some nasopharyngeal swab or whatever, and you look for IgA and you say, is it induced? Or even whatever. You don't have to go through the whole panel of, everything. Now they have a marker. They say, is this marker really elevated in human infection? Right. It's more directed.
Yeah, it's a tricky process because if you just measure neutralizing antibodies, that may go along with something else that's actually doing it, right?
Exactly.
Right, but now you have, you look at complement, right? You do a complementation assay. So whatever that is.
That's your FC vaccine.
Right.
Yeah.
But you look, right, so you do you look at IgA specific neutralization and then you follow it up or you conjugate it to complement.
Mm-hmm.
Yeah.
But you don't have to panel in the same fashion. Here it's a panel, right?
This is more of an experiment than it was
right. The other way is a test, whether or not your conclusion is consistent through over different animals.
So there's another little bit here in the discussion that I really like. It goes back to the attenuated virus, the RSV-A2 that we were talking about. First of all, they point out that, dude, we didn't even measure T cells, and we know they're there. Okay,
That's right.
And then they say, interestingly, the live attenuated RSV-A2 vaccine positive control, which was used to ensure that the animal model and viral challenge was performed as expected, attained strong protection against RSV when administered proximal to challenge. Despite this stark protective signature, the mechanism underlying RSV-A2 mediated protection remains unclear, but is likely achieved independently of humoral immunity, which is poorly induced by this platform.
Wow.
Okay. So,
Very cool.
there's another whole can of worms. Okay.
No pun intended.
No pun
So
intended.
if you wanted to test that vaccine in people, you probably shouldn't measure neutralizing antibodies.
Probably
right?
not. They say instead altered innate immunity, trained immunity, and or robust T cell responses have been proposed as critical mechanisms in that particular protection.
Right. So
this is even more complicated than is in the paper by a long shot.
It's why there's no vaccine for humans yet.
Wow, we got lucky with SARS-CoV-2, didn't we?
Exactly. I was thinking that earlier. I'm always reluctant to say luck because it wasn't. It's. I like to use the word fortunate because a lot of people worked really hard on this, and they were fortunate that it worked out the way it did. And I think much better than anybody could have anticipated, and for reasons that we didn't understand. That we're just beginning to understand.
I mean, that's the history of vaccinology. I mean, people tested. Do you think that Sabin and Salk really knew whether or not IgA or IgM or T cells or B cells or complement or this, that, and the other thing was important for polio? No, they gave the vaccine and they said, okay, we're going to look at to see how much seasonal acute flaccid myelitis is dropped. And then we're going to call it a day.
Jenner knew all this stuff.
Jenner
was actually from the future. Okay.
Yeah. I mean,
abducted by aliens and super.
So, to be honest, I mean, we got lucky with the mRNA vaccine, but you got lucky with almost every other vaccine that you ever developed because there's none of this data for looking at the attenuated measles vaccine or the mumps vaccine. They made a vaccine.
Go back to Pasteur, Amy. Go right back to the beginning. He didn't know.
That's what I'm saying.
But it either worked or didn't. They were empiricists.
Yeah. Right.
Works, but what we're trying to do now is to figure out why it's working, right?
Right. So you don't need to know why it's working to get it to work.
That's correct.
You don't. But if you want to make more vaccines like we do now, and you can't do a controlled trial anymore, it would be nice to know some parameter that you could say, this new vaccine reaches it, therefore we could license it.
What new vaccine do you want to make that you can't do a controlled trial again?
Well, many of the new vaccines are still being developed, right, for parts of the world that don't have vaccine because we can't make enough. They cannot be clinical trialed because you have a standard of care. You cannot withhold an.
They can't be clinically trialed in the United States. They can be clinically trialed probably in those countries.
I don't know about if that's true. If the world has a vaccine that works, is it fair to withhold it in Bangladesh?
Well, that's what we're doing. That's why there's vaccine disparities.
Well, I got that from Florian Kramer's article on correlates of protection, where he said, you can't do controlled trials in the future of COVID vaccines. So therefore, we need numbers to be able to license. Anyway, whether that's true or not, I don't know. It's not my argument. But
I don't know that. I don't know that applies to other countries outside the U.S. Obviously Europe has different standards than we do because they have different vaccines and they've licensed. They have licensed different drugs and different things against SARS-CoV-2 than we have.
I know that the head of the Vaccine Research Center in the U.S. listens, so tell us if you can do this or not.
So I'm looking at the Moderna pipeline.
Yeah.
And they've got, I don't know if it's this, they've got a vaccine for RSV called mRNA 1345. Is that the same one that's being used here? I'll have to look back. At any rate, it's in phase 3 trials in adults in the U.S.
Wow, cool.
Yeah, but notice that it's in the U.S. It's no place else.
Oh, no, I'm sorry. My bad. Thank you, Amy. It's worldwide. Phase 3
What's the
trials
number?
worldwide. Excuse me?
What's the number of the vaccine again?
1345.
Nope, it's not the one in this paper.
Okay.
I just searched for 1345. I assume I would pick it up. All right. That's cool. Good stuff. Thank you, Amy, for those papers.
It's good.
Yes.
Yeah, this is great.
Let's do some picks. Dixon, what do you have for us? Another comedian?
No, I'm done with the comedians. They're all finished. That was the last hurrah. What I have now is sort of a mood piece. Okay, so what I want you to imagine is that you've just booked an international flight, and it starts out at around 6:30 at night, and you won't get to your destination until the next morning. And you've had a good night's sleep already, so you're not going to sleep on this flight probably to get used to the time zone of where you're going. And what you're going to do is you're going to pass over a lot of the world, and you're going to look down at that world out of your window, assuming you have a window seat. You've had your dinner. You might have had a glass of wine or so, and you're feeling very mellow. And all of a sudden, you encounter the lights of the evening. And this is a sort of a poetic look from outer space at cities at night, as seen through the International Space Station, but also through lots of other orbiting, all of the Columbia shuttles and all those other space shuttles that they sent up into outer space for various purposes. Everyone got a look at the way the Earth looks at night. And there's something mesmerizing about it, because you start to see patterns in cities that there are no two alike. It's like looking at a series of different species of spiders that have woven these beautiful luminescent webs. And they all have different parts, but they all have central features as well. And one of those central features is the fact that most of the roads that are put down by planners over the centuries, sometimes, depending on how old the city is, they all lead to the center, commerce center of the city. And it's one of the big features that they're trying to tease out of these images to try to gain some patterns and insights into how human beings live in urban settings. And what better way to do that than to look at the way the city lights up at night? So I thought I would share that with you, because you can peruse this. And there's also some features on some of the related sites to this where they give you a unknown city, and then they give you 7 or 8, 9 choices in a strip that goes above that image, and then you have to pick the one that matches the city that they're looking at. And it's startling to see how much variation there has been. I mean, there's no two alike. That means that every city is unique, and however many there are thousands of cities if you want to define all the way down to 100,000 people. And it's quite amazing to see how humanity has occupied the surface of the earth over time. And that's a bit of a philosophical point that I was just dwelling on in my own machinations when I'm sitting wondering, about some of the parts of the book that I'm writing right now called The New City. So these images that I'm sharing with you have a role to play in predicting the way cities will eventually end up looking in the near future and in the distant future. I think you will enjoy this. This is just I'm not going to say anymore.
Dixon, these are the tech behind. These are time lapse photographs, and they're made possible by this guy Don Pettit, who, from spare parts, he built
That's right.
a barn door tracker, so
That's right.
the camera can
That's
move.
At exactly the same rate that the space station is moving. That's right.
And there's a picture of him. He found just parts, and he built this
Yeah,
thing.
yeah,
And now
you're-
it's been
you're
used to
absolutely
take. And there's a picture where there's an electric screwdriver
Yeah,
drill
I
just
know.
floating.
I know.
Those things are pretty heavy.
Yeah, indeed. Not up there, they're not.
And here's the cool part. Some of these can get a resolution of 10 meters.
Exactly.
Can you imagine? You can
Exactly.
look at some things that are 10 meters apart. Wow.
From
Very cool.
250 miles up.
That's very cool.
So one of their featured photographs in the link that you picked was Chicago.
Yeah.
And I remember flying over. This is seared into my memory.
Yeah, me too.
Flying over Chicago one night and seeing this. It's
You
really
can't
impressive.
mistake Chicago. It's
The other one that I'm looking up here, but it doesn't have, at least on the website, the resolution that I would like is London.
Oh, right.
London's interesting because that, evolved as two different city centers
Indeed.
that ultimately emerged. So I can't tell in the picture that I can pick out here. There's ultimately, it looks like a plaque. There's only one focus in the end, I think.
That's right.
But the resolution isn't good enough for me to tell exactly where that is.
Cities at night are plaque essays for people.
Yeah.
Yeah, it's a plaque of people. Very good.
So the other city to look at that's really fascinating is Paris. And if you go on Google Images and just type out Paris seen at night from space, you will get the most amazing high-resolution picture of Paris. And you can almost see the people eating their croissant.
Yeah.
It's quite amazing.
Nice. Thank you, Dixon.
You're welcome.
Amy, what do you have for us?
So initially it was one editorial, but there's been several in the Times that I think are pertinent and interesting and really very poignant. So the first one that I read this past week is written by an ER physician in rural Michigan. And we discussed it or alluded to it the other night, and people yelled and screamed, and it was like, don't call Trump supporters unvaccinated. And I'm not calling anybody unvaccinated. He calls people unvaccinated. So he's an ER doctor, and he talks about his experience because the hospitals that he visits or services are completely full. And the hospitals within basically a 2 to 3 hour radius of him are completely full. And the selfishness of the whole situation and stuff. And he points out, he has all these people who are dying, and they're all unvaccinated. And they probably have reasons to why they are unvaccinated that whatever, don't really make any sense. And he talks about how nobody has factored in basically the stress that the medical field and the other people in the world and your community go through when you decide not to be vaccinated and you cause these log jams in the hospital. So he talks about how his best nurse drives home crying every night because she's overworked, stressed out, and, sees all these people dying. But it's not just from that. It's also the inconvenience of the people who are your neighbors and your friends who break bones and have heart attacks, and how their care has been completely compromised. And even members of your own family's care has been completely compromised because the physicians are so attentive to, and COVID takes up so much of their time and energy and space and equipment in the hospital, that these people all get subpar care. And at the end, it's saying that your life is more important than somebody else's. And you should never say that your life is more important than somebody else's. And so I think it's a point of view that is really something that people have forgotten and overshadowed with the idea of, well, I don't want the government telling me what to do. It's my liberty of what you do to my body. Sure, it's your liberty of what you do to your body. But at the end, if you break a bone, you're expecting the doctor to drop everything at the ER and put a cast on your bone. And he can't do it because somebody else has taken the same position. I'm not getting vaccinated because I don't want the government telling me what to do with my body, or some other stuff, and people don't think about it at all in these terms. They just look at it very unidimensional, and it's not. And then there were several others, but one that came out this morning was by a virologist at Hopkins, which basically the only part that's important of it is the very last paragraph, which states, in December 2019, SARS-CoV-2 entered the human population and it had no immunity to it. In December 2021, the Omicron variant is entering the human population that has a large amount of immunity to SARS-CoV-2. That immunity in and of itself will lessen the disease severity of the variant. And then he goes on, whatever, but the last sentence basically is that COVID will become a milder disease, not because of the decision that the virus makes. It's a decision that all of us make when we take advantages of the vaccine. So I think those are the lessons that people should, or those are the topics that, or ideas that people should think about in context of whether or not they vaccinate their kids or themselves or whatever.
This article that you picked of the Michigan thing has an embedded video called "Dying in the Name of Vaccine Freedom."
Yeah,
that I've only started yet, but I look forward to looking at that.
yeah.
There's one segment on the PBS NewsHour every night about this. Last night,
Yeah, and they had a Michigan doc on last night, right?
They did.
Dixon?
And they also had one on from Vermont. And Vermont has a 75% vaccination rate. They're the highest in the country in terms of vaccination rate. And they still don't have enough for herd immunity.
Yeah. And I mean,
and people are still not getting vaccinated.
Right. And I mean, I wanted to go to medical school, but I didn't want the responsibility of people's lives in my hand. And now now by not getting vaccinated, you have not only the responsibility of your life, but you're taking the responsibility of somebody else's life. And some of those, like, I knew about the video, but it's kind of like not my thing to watch these kind of things about death. But yeah.
Amy, I hate to tell you this, but there's no solution to this problem.
Well, no, I know that there's no solution, but I think that
it's just a way of stating the problem, but it doesn't suggest a solution.
I think that talking about it loud and clear as much as possible.
Yeah,
right. And I think that
is as much as we can do and probably worth something.
If people don't listen to us, it's because they don't want to listen to us, and therefore.
Right. I don't necessarily know that it is a solution to the problem. I think it's another way to look at the problem in a context that people can understand on a daily basis and say, oh, maybe, so, some of the arguments are, I'll be strong enough to fight off COVID and COVID won't, I live in a COVID-free zone, or whatever. But when you put it in the context of, well, if you needed to go to the city and get treatment and, you need to go for your chemotherapy and you can't go for your chemotherapy because the hospital can't house you because their beds are full of people who have COVID who didn't get vaccinated, maybe you would think twice about whether or not that's something that you should do, because now somebody else who made the same decision is inconveniencing and putting your own life at risk.
Yeah, it's a really good perspective, I think.
Instead of yelling at people and badgering them and calling them selfish and demonizing them, it's a way to address the question in a context that people can usually understand. When it affects them, they can usually understand something. When it affects your neighbor, they just wave their hand, more likely.
Rich, what do you have for us?
On a lighter note.
Or a dark.
The international, I ran across this, I don't know how, a while ago. This is a website called the International Dark Sky Association. And this is an association that concerns itself with light pollution and what to do about it. But one of the things that I really like is that if you look under there's a tab called Our Work, and the first listing there is International Dark Sky Places. They talk about, there's several different links for areas where you can find dark skies. One of which, number 2, is International Dark Sky Parks. So there are areas that are dedicated to where there's not a lot of light pollution, that are dedicated to keeping it unpolluted, where you can go and look at a good night sky. There's even down, if you scroll down a little further, they have an interactive map. So this is a map of the world that shows where all of these places are, and you can search it. You can zoom in on your own place and see where there's good night sky viewing. And I think about this when I grew up in a light unpolluted space, or what was light unpolluted at the time, a fairly rural community in north of San Francisco in Marin County. And during the summer, I used to put a lounge out in the back and sleep out in back in a sleeping bag, staring up at the sky, and you could see the Milky Way every night, and a whole load of shooting stars every night.
And no satellites.
My grandchildren, my grandchildren don't know what the Milky Way looks like.
Exactly.
Because of the light pollution. So I'm, among other things, looking at this map and saying, wow, let's go to Fredericksburg or Wimberley or something like that and get a good view of the unpolluted night sky. So there you go.
Rich, when I was on sabbatical, I had the privilege of traveling in Australia, which I was stationed in Melbourne. And I flew to Alice Springs and then took a tour to Ayers Rock. And there was a hotel at Ayers Rock, and we stayed there for 2 days, and we climbed the rock, which you're not allowed to do now. Then we got back in the bus and went back to Alice Springs, and then I flew back to Melbourne. The bus driver, which drove us at night from the hotel at Ayers Rock back to Alice Springs, stopped halfway, which was about, maybe it was 200 miles or something. And he got everybody out of the bus. He had to wake us all up. And he said, I want you to look up now. And we could see virtually every star at the end of the Milky Way. We saw the end of the Milky Way, the greater and lesser Magellanic Clouds. We saw the Southern Cross. I remember it was like you were looking at in a planetarium with perfect vision, with perfect stars, with perfect everything. And he said, you'll never see this again like this because of the light pollution.
Right.
But what an amazing, is that one of those spots that
I'm just looking here. There's looking out from Melbourne, there's a site called River Murray that's just northeast of Adelaide. There's another site called I'm going to appropriately bungle this. It's called the Warrumbungle National Park. That's actually significantly north of Canberra. And then up in way north of that, in Queensland, there's another spot called the Jump Up. But I can't believe that Australia isn't lousy with good viewing places. Those are the only 3 listed here.
Yeah, it must be.
Yet this is completely the antithesis of your pick, Dixon.
Exactly.
Where I can go to space and pick out every city.
Right.
Yeah, exactly. Yeah.
That's right.
And if I go up far enough and I know where Paris is, I can see all the way down to people eating their croissants late at night.
Yeah, it's the flip side. Dixon was up looking down. This is down looking up.
There you go.
No, just even in terms of light the light and the embracing of the light. So here you're embracing the darkness, and you're saying
the other thing that you get from at least my perspective is that look at all the energy we waste. Those lights are on all night, even though people are not taking advantage.
Yeah, I think about that all the time.
I do too.
Yeah. I'm just saying that it's just.
I shut the lights off here before I leave.
I'm just saying that it's just very different.
Yeah.
That your picture, like,
Yeah.
the humor of it is not lost.
It's ironic. I think it's ironic. And, Rich and I have similar mindsets. You grew up around Marin County. I grew up around San Lorenzo, which is north of Oakland. So we had similar, ecological settings for our early youth.
My pick is a webpage called SARS-CoV-2 Variants. It's put together by the IDSA, the Infectious Diseases Society of America. There's a very nice summary of the variants. It starts with Omicron. It has links to information. In particular, you can find all the papers. The four manuscripts that are out so far are all listed here. And then if you go down further, it has a table of clinical data for each variant saying, for alpha, how do you treat it, what vaccines, et cetera. So kind of a nice site that puts a lot of information together that you might want to access at once.
Yeah, I really like this.
So they're going to collect it all for you here. We have a listener pick from Keith. Enjoying the show. You do such a good job at keeping us informed. Here's a site you may consider for a weekly pick. It's called Ventuski. Never wonder about the temperature, pressure, humidity, snow cover, or any other aspect of weather. Keith is from Kelowna, Canada. And this is, it's a beautiful map, and it's got everything you'd want. Temperature, precipitation, radar, satellite, clouds, wind speed. They actually have the wind on this map, so you can see the prevailing winds wherever you're looking at. It's nice. Thank you. Thank you very much, Keith. That's TWiV 841. You can find the show notes at microbe.tv/TWiV. If you have a question, a comment, or a pick, send it to twiv@microbe.tv. For the months of November, December, and January, if you donate to Parasites Without Borders, they'll match your donation and send it back to us, parasiteswithoutborders.com. And if you'd like to give $1,000 or more, I will send you free an autographed copy of Principles of Virology, 5th edition. Dixon Despommier is at Racaniello.org, thelivingriver.com. Thank you, Dixon.
Quite welcome. And the lights have just been turned on in New York.
Is that right?
Yep. I can look out now and the George Washington Bridge is all lit up.
Very nice.
I don't know. Not sure.
Well,
I must have too much light pollution.
Rich Condit, an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. Get vaccinated.
Amy Rosenfeld is an assistant professor at Columbia University. Thank you, Amy.
Thank you.
You can find her at enterovirus.net.
Yes.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.